Hello everyone. Today we will talk about Cosmid. Cosmid are basically a combination of a plasmid and a lambda phage. Right? It is chimeric in nature. Right? It means that it has both the regions. Right? Region from the bacterial plasmid and the region from bacteriophage lambda. Right? As you know, this is the typical structure of Cosmid. Here we can see this is the lambda DNA site, and the rest of this is the plasmid, bacterial plasmid DNA. So it is a combination of both the part, right? So we will talk about its structure a little more. And as we see, it has cos sites which comes from the lambda DNA. Just after cosmids enter into the host cell, they adapt the circular form because of this cos sites, right? So what I'm trying to say here is, suppose this is the cosmid. There is no particular shape of it. Suppose it is like this, right? But when it enters the host cell, this is our host cell, right? When it enters the host cell, because of the cos sites which are present in the lambda phase part of the cosmid, what they do is they adapt a circular part inside the host cell. The moment they enter the host cell, right, they adapt a circular form. So and they are hybrid in nature between a lambda phage DNA molecule and a bacterial plasmid. Their designs, due to which their designs are based on the fact that the enzyme that package the lambda DNA molecule into the phage protein coat, that is the capsid, only the cos sites in order to function. They only need the cos sites in order to function, right? So what I'm trying to say is, when we package those enzymes which are present in the bacterial plasmid or the lambda DNA molecule, which we package into the capsid, right? Capsid are what? Phage protein coat. So during this time, they only require what? Cos sites in order to function. There is not any requirement of that. Now, in vitro packaging, we know what happens in vitro packaging. It works not only with lambda genome, but they will work with any molecule that carries the cos sites. All right. So what we are saying that in vitro packaging, right? In vitro packaging, what are the conditions it has shown till now? In vitro packaging, not many conditions it has shown, right? So in vitro packaging, there are only two conditions. It doesn't matter whether it should only be the lambda genome, no, but it can be it can work with any molecule, right? It can work with any molecule as long as that molecule has what? As long as that molecule has cos sites, right? What sites? Cos sites. And do this cosmids have cos sites? Yes, definitely they have cos sites. And this cos site should be separated by a segment or a part of DNA of about 37 to 52 kilobase pair. DNA should be separated between this cos sites. It should be separated by this many length of DNA. So cosmid is basically a plasmid that carries the cos site. I can say that, right? Basically a plasmid that carries the cos site. It is a very simple definition. We should remember it, and always we should remember it. So what I said was, cosmid is basically cosmid basically a plasmid that carries cos site. And where does this cos sites come from? Obviously the lambda phage. Due to this cos site, we just learned that there is in vitro packaging, right? Now cosmids also have a unique multiple cloning site regions into which the DNA fragments can be ligated, right? Unique sites cosmids have, like eco R1, and just like we saw BAM H1, right? Here we saw BAM H1, I think. It's BAM H1. Like it has BAM H1, right? There are different multiple cloning sites, unique cloning sites. Now it also needs selectable markers such as the ampicillin gene. What we talked before was about the multiple cloning sites. We are talking about selectable markers such as ampicillin resistant gene, right? Ampicillin resistant gene signified by this symbol, and obviously an ORI, that is replication origin, right? These both two things are obviously necessary for a Cosmid. And another, what was another thing? Multiple cloning sites. These three things are very necessary for a Cosmid, and definitely yes. How can you forget it? The cos sites, right? Cos sites, and all these things are definitely very necessary to make up a Cosmid. So we will talk about the cloning procedure. How's the cloning procedure in Cosmid, right? Cloning procedure is almost the same as lambda bacteriophage, right? In Cosmid, and it has when less than 48 kilobase pair of foreign DNA can be carried by Cosmids. How much? Less than 48 kilobase, right? Of foreign DNA it can carry by a Cosmid as a vector. And the presence of the cos sites obviously enables the enzyme produced by the actual phage to package it into a phage capsid. And cos sites again of help provides cos sites provide it, the enzymes which are necessary which to be packaged into a phage, right? And then Cosmids have disadvantages too. It has two big disadvantages. Cosmids have two disadvantages. Anything out there definitely have some disadvantages, right? The first disadvantage, what is the first disadvantage? Is the high frequency of recombination inside the bacterial host. High frequency of recombination, high frequency of recombination inside the bacterial host, right? It means that it can easily combine, it easily can recombine with the bacterial chromosome. Suppose the Cosmid is there and it enters the host, and it may recombine due to some similarity or some remarkableness between those bacterial hosts gene genetic material, the genetic material of bacterial host, and between the Cosmid, to which recombination may occur, right? And Cosmids are unstable inside the host cell, thus easy to lose the vector, right? They have highly unstable inside the host cell. And when we are talking about this disadvantage, we are talking about when they are already inside the host cell, right? Not normally. When Cosmid enters the host cell, it may recombine with the genetic material of the host cell, and it is obviously unstable during that time, due to which it may easily lose its function as a vector. Now, we will talk about the cloning experiment with a Cosmid. A Cosmid is opened at its unique restriction site, right? Like BAM H1 we talk about. So we will see it. Here it is, right? Cloning experiment. We are considering an example of Cosmid which is PJP8, right? Now, in this, we see you can see that the Cosmid is opened at its unique restriction site, right? What is the unique restriction site of this Cosmid, and which is BAM H1, right? If you can see when we restrict with the restriction endonuclease BAM H1, and so you can see this linear Cosmids are formed after the action of this endonuclease. And these fragments are usually produced by partial digestion, right? We should remember this partial digestion, because total digestion will obviously lead into the fragmentation too. It will lead to the fragments to be too small to be cloned with a cosmid, right? If total digestion is done, then this fragments will be too small to be cloned with a cosmid, obviously, right? And here we can see when then they are ligated with catenins. Catenins are called chains, right? Chains. As you can see, there is a chains of this particular part, 1 2 3, 1 2 3. As you can see, 1 2 3, they're continuously going on like chains, like chains. They are joined in a chain form, 1 2 3 again, they are joined, right? And this new DNA bands are inserted in this between the cos sites and the resistant gene part, right? This is the resistant gene part and this is the cos site. Here it is restricted, and here it is already BAMH present, and then this comes, right? This becomes, and this comes, and between this cos and ampicillin resistant gene, they settle down, right? And ligates, right? Ligation is carried by ligase. Then in vitro packaging, we already learned because of the cos sites. Now we assume that the inserted DNA is of the right size, right? I just told you that inserted DNA should be undergone partial digestion, not complete digestion. The inserted DNA that we talked about, that we are inserting here, that this inserted DNA, it should be undergone of right size, only partial digestion, not complete digestion, because complete digestion will make these fragments too small to be carried with cosmid, right? Only partial digestion with the however we are doing it, right? When this enter, and we are assuming that the inserted DNA is of the right size, what it happens, right? What in vitro packaging happens is that it cleaves that cos site, right? It cleaves that cos sites, this sites, it cleaves them, it cleaves them, and then it packages this the rest of that recombinant cosmid into the mature phage particles. This is the mature phage particles, right? And when this mature phage particles are made to infect an E. coli culture, plaques are formed, as you can see here. It infected the E. coli cell, and then colonies containing circular recombinant PJ 8 molecules are formed in an ampicillin medium, right? And